Hello, dears, and welcome to Al-Hussaini Virtual Lab Pathology Talks, Tips, and Practical Tips. What I'm going to be sharing with you today is a nice example of supratentorial midline pilocytic astrocytoma. So this is a case of a 10-year-old male patient who was found to have supracellular brain tumor or brain mass. And this is the low power magnification of the fragments that were submitted for examination from this tumor. And as you can see here from the low power magnification, the tumor is kind of pink in color, which gives you a hint that this is probably an astrocytic tumor. There is no tendency for any perivascular rosette formation. The tumor is not showing any increase in the cellularity or a bluish color, which would rule out in a way that it is an embryonal tumor. This is on higher power magnification, and as you can see, there is a proliferation of those kind of comma-shaped or oval-shaped nuclei set in a fibrillary background with some hint of vascular proliferation. This is a higher power magnification, and as you can see here, we have the nuclei of the tumor cells, and then we have the processes in the background. Now, mind you, if you pay attention, there is no evidence of Rosenthal fibers nor eosinophilic granular bodies, which are typical features seen in pilocytic astrocytoma, in particular if it was in the posterior fossa, but also, of course, supratentorially, if it shows the typical features. However, remember that myxoid astrocytoma, which is now a subtype of pilocytic astrocytoma, typically does not contain these structures. This is another focus where we can see the proliferation of the cells. These are the oval-shaped nuclei and many elongated processes in the background, yet with no evidence of Rosenthal fibers of eosinophilic or eosinophilic granular bodies. And one would start to wonder whether this might be an infiltrative astrocytoma actually. Here we can see the tendency for some microvascular proliferation. But if this was an infiltrative tumor with microvascular proliferation, then we're kind of heading towards grade 4 astrocytoma. But actually, this is the case. This is not the case, and this is where the tip really lies. So let's move now into the immunohistochemistry. Now, GFAP is strongly diffusely positive, and in a different context, in a different setting, we might think of microvascular proliferation highlighted with the GFAP immunostain. Now, this is the neurofilament protein, and I meant actually to do it and to show you, to share it with you, to highlight the infiltrative nature of the tumor. So the tumor does infiltrate pre-existing structures, and this is another focus where we see really the axons of the neurons, the neurofilament protein, and then we have some, again, what appear to be a microvascular proliferation. However, remember that this is a supracellular mass in a young patient, and we have always to keep pilocytic astrocytoma in the differential diagnosis. The IDH1 immunostain was completely negative. ATRX showed retained nuclear staining in the tumor cells. These are the strongly staining ones are the neurons. So neurons are kind of entrapped within the tumor. And then we have the lightly brown tumor cell nuclei, which means there is retention of the ATRX immunostain. P53 showed this mosaic weak staining pattern, which goes with wild type staining pattern. And because the tumor is kind of midline, I elected to do the H3K27 trimethylated. And as you can see, tumor cell nuclei shows retention of the nuclear stain, which means that the tumor here is non-immutant. Now, since we thought of pilocytic astrocytoma, we did the immunohistochemistry for BRAF to look for the mutation. And this is kind of difficult actually to interpret because of the presence of this granularity, even on the, including the endothelial cells of the blood vessels. So this was interpreted as non-contributory. Now, what we did also, we did methylation studies on the tumor, and the calibrated score came back as pilocytic astrocytoma, supratentorial midline pilocytic astrocytoma with a high score, meaning that we're confident of the diagnosis. The copy number variation, interestingly, showed that fusion with the KIAA11549 with the BRAF in support that this is a pilocytic astrocytoma rather than an infiltrative astrocytoma or a midline tumor. So the final diagnosis in this case is actually pilocytic astrocytoma, CNS WHO grade 1. What I wanted to share with you in particular with this case is that sometimes the pilocytic astrocytomas, especially supratentorially, sometimes also in the cerebellum, might show this infiltrative growth pattern in the absence of Rosenthal fibers or eosinophilic granular bodies, or the typical biphasic morphology or growth pattern that typically seen with pilocytic astrocytoma. This does not negate the diagnosis. Pilocytic astrocytoma can sometimes show this diffuse growth pattern, and which was further highlighted with the neurofilament protein. Now, the presence of other worrisome features like the microvascular proliferation loses its significance if we talk about or in the context of pilocytic astrocytoma. So before we assign this tumor into a grade 4 because of the infiltrative growth pattern, because of the absence of the typical features of the pilocytic astrocytoma, and because of the presence of the microvascular proliferation, remember that supratentorial tumors in children are likely to be pilocytic astrocytoma, which might show the diffuse growth pattern, so that this is the important pitfall that we should pay attention to. I hope you find this tip useful in your daily practice. Thank you.